students, welcome to the Biological Life Sciences channel. In today's video, we are going to discuss the DNA organization in prokaryote and eukaryotic system with few examples. So let's start. First of all, we are going to see organization of DNA in prokaryotes. In prokaryotic cell, a single cellular DNA is present. Circular DNA is present, but recent research has also shown that there can be even multiple or linear forms of DNA. It's relatively smaller in size in prokaryotes. They lack any membrane-bound nucleus, thus the DNA is present in nucleoid form. The replicative form has two entangled daughter DNA molecules separated by topoisomerase enzyme, which is called DNA gyrase. There is no problem of end replication as in eukaryotic cell. End replication is a problem there. Here it is easily replicated, and there is a presence of extra chromosomal circular DNA also, that is called plasmids. The DNA here is not associated with any kinds of histone proteins, and there is a presence of a single origin of replication in case of prokaryotic DNA. It also lacks centromere and telomers. So let's see before gene density, before we go to the next topic, that is, it is average number of genes per Mbps of DNA. The 4.5 Mbps genome of E. coli, when studied, showed that it comprises almost entirely of genes, that is 4400 approximately, except for a small region called ORIC site, that is origin of replication, which does not code for any protein but marks the site for origin of replication. The gene density has been calculated to be 950 in case of E. coli. So the gene density is very high in E. coli, as we can compare it with the eukaryotic cell, where there is a presence of lots of non-coding intertwining sequences. So therefore what happens, the density of the gene is very less as the genes are apart from each other. So in humans, nearly tenfold it is lower, that is 9.3 gene density is present in humans. All other results shown that an inverse correlation between gene density and organism complexity, that is, the more complex the organism, lower is its gene density, while the simpler organism, the higher is its gene density. So what causes this gene density to be higher in prokaryotes? The non-coding regions or intergenic sequences like introns in prokaryotes are absent. So all the genes are linked together continuously, there is no introns present within the genes. Repetitive DNA is negligible in eukaryotes. As in eukaryotic cell, there is lots of repetitive DNA sequence present, but in prokaryotes there is negligible repetitive DNA sequence. And one more thing, there is a presence of polycistronic genes. So what will happen, 2 or 3 genes will be fused together in the form of operons in prokaryotic cell. So for 3 to 5 genes there will be presence of regulatory sequences. So it will reduce the load on different genes. So ways in which prokaryotic DNA differs from eukaryotic one. First of all, its quantity, that is in prokaryotic cell it is much lesser and eukaryotes much higher. Replication, it's more complex in eukaryotic cell than in prokaryotes, and the information content and its packaging, it's much simpler in prokaryotes, whereas it is highly complex in eukaryotic cell. So let's see the quantity first. Prokaryote typically have one main chromosome, though it might have few copies of it. These cells can also have smaller DNA structures called plasmid. It has additional information. The number of nucleotide base pair present in prokaryotic cell ranges from 160 to 12.2 million species wise, means it is different in different species. Eukaryotes frequently have multiple type of chromosome with many more base pairs as compared to prokaryotes. For example, humans have 23 pairs of chromosomes. The maternal and paternal sets constitutes of about 2.9 billion base pairs, but here it is 1 or 2 chromosomes only. Let's see how it is packaged in prokaryotes. A prokaryotic chromosome is circular, resides in the cell region called nucleoid. So in nucleoid there is a presence of DNA. The types of protein found in prokaryotic chromosome is known as nucleoid associated proteins, that is different from histones that is appears in eukaryotic chromosome and causes the prokaryotic chromosome to form a looped structure. Unique eukaryotic chromosome packaging features includes tight coiling, dense packing, enclosure with a nuclear membrane linear than circular DNA. Let's see the different characteristics of prokaryotic chromosome. It is located in nucleoid region, not nucleus, since nucleus is not present here. It is single-stranded, double-stranded, covalently closed circular DNA. It is approximately 1300 micrometer in length. Typical prokaryotic cell is about 1 micrometer in length. The center of the chromosome is attached to the plasma membrane of the prokaryotic cell. This helps in partition the chromosome during cell division, and it is negatively supercoiled. The DNA is arranged in series of loop here. Each loop is supercoiled. The loops are attached to the proteins. Attachment to the proteins prevents rotation of past loops, means it prevents entangling of DNA sequences. The proteins are basic here, positively charged, allowing them to interact with negatively charged DNA, which is there due to the presence of phosphate group. The number of loops can be determined by number of cuts needed to remove all supercoiling. Removing all supercoiling should necessarily reduce S value by 30%. Single-stranded breaks remove the supercoiling from a loop. It takes 45 to 50 breaks needed to obtain this with E. coli chromosome. This corresponds to the number of DNA gyrase binding site. DNA gyrase includes negative supercoiling. See, when in gene expression, this coiled DNA has to be negatively supercoiled. So it is done by DNA gyrase enzyme, and there is a presence of lots of site for this enzyme. The eukaryotic chromosome looks like this. Not all bacteria have single chromosome, but some bacteria do have multiple one. To fit DNA into the bacterial cell, the chromosomal DNA must be compactly arranged about 1000 folds. Protein organizes DNA into loops and looped structures, compacts the chromosome about 10 folds. An additional 100 folds is introduced by supercoiling. So this is continuously supercoiled into the loop form, which forms a DNA like this. DNA supercoiling, there are two types, negative supercoiling and positive supercoiling. Most negative supercoiling occurs during normal growth. See, in normal growth of bacterial cell or prokaryotic cell, it will require lots of genes to be transcribed. So that genes has to be opened up, and this is done by negative supercoiling. Different proteins are involved in supercoiling. HU proteins, that is present in nucleoid, it works with the enzyme called topoisomerase 1 and helps in generating negative supercoiling. And there is a protein called integration host factor IHF. It binds to the specific sequence within the genome and introduces additional bends. The folded DNA is then organized into a variety of conformations that are supercoiled and bonded around the tetramer of HU protein. Once the DNA has been condensed, DNA topoisomerase 1, DNA ligase and other protein helps in maintaining the supercoil in the nucleoid form, as we have seen the picture in the last slide. Because each domain of the chromosome is independently coiled, introducing a single-stranded nick in DNA by the treatment of DNase enzyme, which cleaves the DNA at internal site, will relax the DNA only in the nicked domain. Unnicked domain will remain supercoiled. Means the domain which is nicked will only get negative supercoil, other domains will be as it is. You can see here in this diagram how the supercoiling is occurring and how it is cut by the different enzymes and it is released. See, here the cut is made to release that, then topoisomerase introduces the negative supercoil, thereby loosening the DNA. There is also presence of extra DNA that are called as plasmid other than the chromosomal DNA. You can see here in the bacterial cell, this is the chromosomal DNA and this is circular DNA that is called plasmids. They are independent of chromosomal DNA and they are capable of replicating autonomously within suitable host. Benefit it gives to the bacteria, that is, it provides protection against different antibiotics, therefore it's having antibiotic resistance genes. There can be more than 1000 copies, but in some cell it is limited also, and it codes for the gene that allows to infect the host immune system, that is, various virulent genes are present in this plasmid DNA. Let's study about eukaryotic chromosome. If the proteins are removed from eukaryotic chromosome, it would be about 2 nanometer thick by 12 lakh nanometers long. Let's see histone proteins. Histone proteins are associated with the DNA. These are basic in nature and it contains large amount of cationic amino acids like lysine and arginine. These are all having positive charge, so it attracts the negatively charged DNA. It is arranged mostly in alpha helices. The positively charged protein allows the protein to interact with the negatively charged DNA. The alpha helices fit into the major grooves of the DNA and helps in tightening the DNA. There are 5 classes of histone: H1, H2A, H2B, H3 and H4. 4 of these are present in dimer form, which forms the histone automer in the nucleosome. It is highly conserved one. We can study the detailed structure of nucleosome here, and I'll give you the link in the iCard also. There is only 2 amino acid difference between the cows and peas, which diverged 10 years ago. So histone proteins are comparatively similar in different organisms in eukaryotic cell. This histone protein core octamer forms the nucleosome. This DNA is wrapped around this octamer of histone proteins. It is associated with the DNA referred to as core DNA. H2A, H2B, H3 and H4, these are the 4 histone proteins that are present in dimeric form, and it forms the octamer. Core DNA is about 146 base pair in length. The space between the nucleosome is referred as linker DNA. Linker DNA is associated with H1 that helps in tightening of this DNA wrap around the octamer. The DNA associated with histone has a bits on a string appearance, and it is about 11 nanometer in diameter. This bits on a string fuses together to form a solenoid. The H1 histone will link the nucleosome into a solenoid structure. The solenoid is about 30 nanometer in diameter. This is the state of DNA in resting nucleus. Then it goes to scaffolding. Chromosomes are compact 100 folds more by scaffolding with non-histone proteins. The scaffolding of chromosomes is around 700 nanometer thick. This is the state of DNA during mitosis and meiosis. It helps in also compacting of the DNA genetic material. Let's describe the process of DNA packaging in eukaryotes. Nucleosome chain coils into a solenoid model which is stabilized by H1 protein. So remember, H1 protein helps in formation of solenoid model. Solenoid forms loops and loops attached to the central scaffold. The scaffold plus loops arrange themselves in a giant supercoil that forms highly condensed metaphase chromosome. We can see the DNA here, packaging, how the packaging is done in eukaryotes. First of all, it is converted into a bits on string fashion, then it is converted into chromatin material, then it is chromatin loop, and then condensed chromatin loops are supercoiled together, and then it forms the chromosome, which is around 400 nanometer in breadth. There are different levels of packaging here. First level is nucleosomal level, that is this one, then solenoid level and chromatin level, then looped domains are formed, and then fourth level is metaphase chromosome level. Let's describe the histone here. These are the positively charged protein-rich amino acids with basic R group as mentioned earlier. The core histone forms a complex of 8 histone polypeptides, that is octamer, to form the core of nucleosome around the DNA wrap, and can be chemically modified by different enzymes. So it helps in gene expression also. You can see here in this, there is histone octamer present, and the DNA is wrapped around this, and then there is a presence of linker DNA that links one nucleosome to the other. So this is how the nucleosomal arrangement is arranged in a bits on a string fashion. You can see here the diameter of this nucleosome is 10 to 11 nanometer approximate. It is different in different species. Then this is again supercoiled using H1 histone protein. This one, it is coiled again to form a solenoid model. There is a presence of 2 different regions in eukaryotic chromosome, that is heterochromatin and euchromatin. The heterochromatin part is densely packed. The density of the gene is a bit less here, so it is not readily transcribed, and these will be darkly stained during cell division, so therefore it's called heterochromatin. On the other hand, euchromatin, this DNA part is less densely packed in chromosomes, as this is transcriptionally active, so it has to be unwinded regularly for the gene expression. You can see the diagram of chromosome here. This cohesin protein helps in binding the sister chromatids together, and at the center there is a presence of centrosome that holds the sister chromatids together from both ends. So this is how the chromosome is condensed at metaphase. This is the prophase chromosome and this is uncondensed chromosome. Let's see the whole genome and how it is divided in eukaryotic cell. There are two different types of sets in a particular genome. That is first coding region and another one is non-coding region. The coding region here is divided into two parts, that is coding for genes, that is different proteins are synthesized from this, and other are the sequences, that is either introns that are present in genes or untranscribed regions that are present before and after the gene. The non-coding regions here are intergenic sequences that are called microsatellitic DNA. These are small tandemly repeated sequences such as VNTRs, and there are unique regions also. These are non-functional regions, also sometimes referred as pseudogenes. And other one is genome wide repeats. This may be few to many copies, also called as transposons. So let's see the comparison between prokaryotic and eukaryotic chromosome. The chromosome structure in prokaryotes, it's mostly circular, but in some cases it's linear also, but in eukaryotes it's linear. Chromosome number single or multiple in pro, while it is multiple from 2 to 50, it's less than 50. Location, it is in nucleoid in case of prokaryotes, and it is in nucleus in case of eukaryotes. Genome size is comparatively about 10 raised to 4 to 10 raised to 7. Here it is 10 raised to 8 to 10 raised to 11. The gene density is very high in prokaryotes while it is low in eukaryotes. Non-coding region is not present or rare. It is usually present. The intergenic sequences are absent or very low in pro, and it is present in eukaryotic cell. The repetitive DNA is very low while it is present in eukaryotic cells. Histone proteins are absent here in prokaryotes and it is present in eukaryotes. Cistron in eukaryotes, it's polycistronic mRNA formed here because multiple genes in the forms are linked together. It is always monocistronic in eukaryotic cell. Origin of replication is single in case of prokaryotic cell, while it is many. It is about 30 to 40 kb apart, and there are many in number. The centromeres are absent in prokaryotes while it is present in prokaryotes. Telomeres are also absent, present in eukaryotes. Chromosomal duplication, it is poorly understood in eukaryotes, but duplication and segregation occurs in distinct phase, that is cell cycle. It is understood clearly in eukaryotic cell. The cell cycle checkpoints are absent in case of prokaryotic cell, as the organism is a very simple organism, but it is present in eukaryotic cell. The site of transcription and translation, it is coupled one, that is as the transcription is occurring, translation also starts, but here in eukaryotes it is not so. The mRNA formed has to be taken out of the nucleus to the cytoplasm where translation occurs. The mRNA, it stays for few minutes only in case of prokaryotes. Here it stays for few hours, even days, before it is destroyed. The promoter region is present in prokaryotic cell. It is generally Tata box, that is -10 base pair -10 element, and RNA polymerase recognition site is present at -35, whereas it contains Tata box -20 to 25, and there is a presence of distinct control element called as enhancer, which helps in transcription. So this was all about eukaryotic chromosome. Now let's see the chromosome of lambda bacteriophage, that is lambda phage. Esther Liderberg found E. coli K12 strain to be lysogenic for lambda phage, which marked the discovery of lambda phage as well as the phenomenon of lysogeny. So it's a temperate phage, it's a lamboid phage that infects the E. coli. Let's see the characteristics of lambda phage. It is a coliphage, that is infects colin bacteria. It belongs to the Spiroviridie family. Its size is nearly 50 nanometer diameter. Isohedral head is present. It is having a flexible tubular protein tail. Connector serves as a site for attachment of the tails to the head. There is a presence of cos site on the either end of the DNA of the lambda-phage genome. It has a double-stranded DNA, linear DNA, that is in B form, and about molecular weight of 31 into 10 raised to 6 daltons. And since there is a presence of cos sites here, it adopts circular structure when injected into the host cell. This is the structure of the lambda-phage. The size of the tail is around 12 nanometer. The fiber is around 25 nanometer. The middle part or tail is 150 nanometer. Then the head is 55 nanometer. Let's see the genome organization in case of lambda-phage. There is a presence of left hand region and central region and right hand region. In left hand region, genes are included such as NU1 through which J that codes for head and tail protein. Central region, this includes RJ through GAM that codes for integration and recombination functions. All these are the genes that are present here that is responsible for attachment of the phage with the bacterial cell membrane. The right hand region, there is a presence of N and Q control that lysogenic and lysis cycle. These four are the gene very important for controlling the lysis and lysogenic cycles in case of this phage. So it is present in right hand region. ONP for DNA replication of this genome, and S, R and RZ for host cell lysis in case of lytic cycle. So this is the genome of lambda-phage here. You can see the head region, tail region. There is a replaceable region also. This region is not required or non-essential in case of the replication of lambda-phage. So in cloning, this region is generally replaced in case of replacement vector and a fragment of interest is added. I will put the link in the I button here so you can watch the cloning done in lambda-phage in this replaceable region. Then there is a presence of different genes that are required for lytic regions or lytic cycle. So let's see the genome organization in M13 bacteriophage now. It's a filamentous rod-shaped E. coli bacteriophage. It belongs to the family Invoviridae. It's a small phage. It has circular single-stranded DNA of about 6400 nucleotide in length, that is 6400 base pairs. During replication inside the host cell, M13 is present in double-stranded replicative form, that is RF form, that is circular form, and during morphogenesis the DNA is converted into single-stranded form, that is during its development and infection cycle it is again converted into single-stranded form. When it is again infected, it is converted into double-stranded form, and its DNA is completely sequenced now. It has dimensions of nearly 900 into 6 to 7 nanometers. Thank you. If you like the video, please press like button, share this video, comment and subscribe. For more videos, press bell icon. Thank you so much.